Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is shift 2 that I am discussing, and I am discussing the part C questions. This was conducted in the year March 2025, and here in this section I will be discussing questions from applied biology and methods in biology. That is your last 2 units, and if you have any doubt, please write it in the comment section. If you want still previous year question papers of CSIR Net Life Science being discussed, you can go to my channel Omni Bioentrance, and in the channel playlist you can select for CSIR Net exams, and there you will find. So coming to the first question, so they have asked a grammar question, I can say. So you have to write whether it is can or cannot in the blanks. Okay. So first question. Codominant molecular markers dash be used for identification of heterozygotes. So whether it is can or cannot, we will see. So this is the gel of this picture. If you see here, this is the gel of a sample being treated with codominant markers, and this is for the dominant markers. From the gel itself, you can see that parent 1 and parent 2 codominant markers have identified the dominant allele, that is capital A, as well as the recessive allele, that is small a. And in the F1 that is got upon crossing these two parents, you can see two bands, and F1 is a heterozygote. So codominant markers, they can be used for the identification of heterozygote. But what if it is a dominant marker? See here, upon treatment of both the parents as well as the F1 with dominant marker, it was able to identify only the presence of the dominant marker gene, not the recessive one as in case of this gel. And in the heterozygote also, the dominance character has been identified, not the recessive. So dominant marker, it will identify only the homozygous condition, the two dominant condition, and not like the codominant marker, which can be used for the identification of both dominance as well as recessive alleles. So one is not correct, the others we will see. Then genome-wide association studies. Okay, regarding this I have discussed in your morning shift question paper conducted in the same year. You can go through that. Okay. Genome-wide association studies dash be performed on germ plasm with high genetic diversity. Yes, they can be performed on germ plasm with high genetic diversity, because genome-wide association studies, they want large pool of alleles to study the marker linked to the trait. So they choose usually different accessions or different versions of the gene. That is, they can go for, suppose you are taking the fruit color of apple, you can go for the red one, you can go for the green one, you can go for the yellowish red one. All these varieties they can compare. Then you can even compare it with the wild type apples also. So all these things they take into consideration when they are studying the genome-wide association studies to find out the marker linked to the trait of interest. So B is can. So that makes option 2 gone. Then comes to the next one, that is F2 population be used as an immortal population for genetic mapping studies in plants. Now what is F2 population? Here you take two different parental lines, that is if they are differing in disease resistance, or if they are differing in a different fruit size, etc. You take those two parents, you cross them, and you produce the F1 generation. Now these F1 individuals, they are self-pollinated to produce the F2 generation. Now F2 generation, it is used only for initial mapping studies, and it is not designed for long-term maintenance. That means they are a mortal population. What do you mean by this immortal population actually? So what is immortal population is that they can be indefinitely propagated by vegetative means, not sexual reproduction. It is by vegetative propagation that you propagate the plants, and because you are utilizing this vegetative propagation, you do the repeated selfing process. Okay, and you create a lines with the fixed genotype, and this fixed genotype they can be maintained for long-term studies or breeding studies. Okay, so F2 population they are propagated mainly by vegetative means, and they are developed through repeated selfing of the parents. Okay, that is how you develop the immortal lines. Now F2 population, they are not meant for long-term maintenance, so they are a mortal population, and they are not developed by repeated selfing of the parent. So C is cannot. So that makes option 3 also gone. So what is left out? Option 4 will be the answer. Now we will see. Now bulk segregant analysis dash be used for mapping of monogenic qualitative traits. Now what is this bulk segregant analysis? It is also used to identify genetic markers linked to a specific trait of interest. So here also you cross two parental lines, maybe like for disease resistance, some will be susceptible, some will be resistant, like that. So those two, disease resistant and disease susceptible plants, they are crossed together and you create the F1, then you self the F1 and you get the F2. Now you select individuals with extreme phenotype, that is here we are telling about disease resistance, so you select the ones which are highly resistant as well as those ones which are highly susceptible. So highly resistant you will get some plants, highly resistant also some plants you will get. Now you isolate DNA from these highly resistant plant and highly susceptible plants. Then you pool the DNA, that is called as the bulk. Okay, you pool the DNA, that is you pool all the DNA what you got from all the plants which are highly resistant. Then you pool all the DNA from all the plants which are highly susceptible. So you got two DNA bulks, one from resistant plant and the other from susceptible plant. Then you genotype this bulk, that is by using markers like SNP, microsatellite, AFLP. Then you identify the markers that are linked to the trait of interest. So that is what you do in the bulk segregant analysis mapping technique. So they are telling bulk segregant analysis can be used for mapping of monogenic qualitative traits. Yes, they can be used for mapping monogenic qualitative traits. Monogenic qualitative traits, they are Mendelian traits and determined by single gene, and they have only two alleles. That is what is meant by monogenic. And qualitative traits means they do not show the discontinuous character, but rather they show the discontinuous character and do not show the continuous difference in the character, as in quantitative or polygenic inheritance. That is the difference between qualitative trait and quantitative trait. So just for your information I told you. Okay, now so D will be, it can be used for mapping monogenic qualitative trait, so that makes answer 4 as your answer. Then next one, a student performed an ELISA to detect anti-ovalbumin IgG in serum sample. Okay, so anti-ovalbumin IgG in serum sample, that means that is your antibody. The experiment involved the following sequential steps: coating plates with ovalbumin, so that is ovalbumin is your antigen and anti-ovalbumin IgG is antibody. So he or she want to detect the presence of this IgG antibody in the serum sample. Then what they did, they blocked with BSA, then they added serum sample, then they added the anti-mouse IgG HRP, that is secondary antibody conjugated to horseradish peroxidase and enzyme. Then they added the substrate, that is H2O2 and OPD, that is phenylene diamine dihydrochloride, and adding H2SO4. So the student made the following statements. Now before coming to that, I will tell you what is the exact procedure in which ELISA is being conducted. So here first what you do is you coat the microtiter well of a microtiter plate with a capture agent. So in this case it is the antigen that is ovalbumin, which is the antigen that you are coating. So this is the well of a microtiter plate, and these here round what I have drawn is your antigen. Then after adding this antigen, that is your capture agent, you block the wells. Okay. Now why is this being done? This is to prevent non-specific binding of other substance in the sample. That means when you add your sample, it will not be 100% pure. There may be some other substances also which has a tendency to bind to spaces in between these antigens, okay, or on the walls of the microtiter plate. So they will have a tendency to bind on all these places. That is, they are the free sites which are available in the well. So they will have a tendency to bind on these free sites. And in order to prevent that, you add what is called as BSA is usually added as a blocking agent. It will go and bind in these free sites and prevent the blocking of these unwanted substances there. Now if you do not block, it can affect your interpretation of the result as well as give inaccurate results. Okay. Now next is you incubate the sample containing the target substance. So here you incubate it with the sample having your antibody of interest. So antibody against the ovalbumin, that is your primary antibody, it is being added and it will go and bind to these antigens which are coated on the well. Then after that you do the washing. Now each step has got its importance. Listen carefully whatever I am telling, because the options whatever or statements what they have given is important. If you want to understand that, you should understand why in each step you are doing this blocking, why you are washing, all these things you have to know. So after incubation, that is after adding your sample, the sample whatever antibody is there, it will go and bind to the antigen and then you wash it. Why do you wash at this stage? So if the wells are not thoroughly washed, there may be some unbound antibodies, or it can be any other unbound substance which will be floating here. You are adding the solution. So those unbound substance, they will be running up and down here as they don't have anything anywhere else to go and bind. So they will be floating there, and these, or it can be some extra antibodies which don't have sufficient antigens to go and bind. So all these extra antibodies or some other unbound substances present in your sample will be floating there. So in order to remove those ones, you do the washing, or else what will happen? Suppose there are extra antibodies which are floating, then in the later steps when you add the secondary antibody, the secondary antibody will have a tendency to go and bind to these floating antibodies also, that is which are unbound antibodies also, and it can hinder or give inaccurate results. So in order to prevent that, you do the washing at this step. Then comes the detection, that is you add your secondary antibody, that is antibody conjugated with an enzyme. So this is your secondary antibody, it is conjugated to HRP. Here it is anti-ovalbumin IgG conjugated HRP. HRP is horseradish peroxidase. After that again you do the washing. Now why here washing? There may be some secondary antibodies which are not bound to the primary antibody. They will be floating freely there. and when you try to detect after adding substrate, you try to detect, you get more coloration in some wells. In some wells there will be less coloration, like that difference can happen. So if you get more coloration, that means your secondary, you have not done the washing over here at this stage after addition of the secondary antibody. So if extra secondary antibody is present, that is which are unbound to the primary antibody, they can also react with the substrate and give a signal, which can result in improper interpretation of the result or inaccurate results. So in this stage also you will do the washing. Then you add the substrate addition. So the substrate, it will depend upon what enzyme you have conjugated to your antibody, and the enzyme reacts with the substrate and it produces a detectable signal, and usually it is a color, and the intensity of this signal can be visualized or measured using a spectrophotometer or any other appropriate instrument whatever is available. And the intensity of the signal, it is directly proportional to the amount of the antibody, or if you want to find out regarding the antigen in the sample. Okay, now come to the question. Okay, now before reading all these things, you can see here they have mentioned specificity, sensitivity, specificity. What is all these with regards to ELISA? Now suppose you want to identify individuals who have the disease. So what you do is you plate it with the antigen, then you add the serum sample which will have the antibodies, and you find whether these antibodies are bound to the antigen. So in that way also you can detect diseases in patients. So what is sensitivity means? That is, they will identify the true positives. That is, it will correctly identify individuals who have the disease or the condition. That is sensitivity. Sensitive, it gives a sensitive result, means it will give only those result of those individuals who are really positive, really having the disease. They will be identified properly with ELISA. That is what it means. That is sensitivity. Now sensitivity can be affected by if you are not washing it properly. That is, at each stage if you are not washing, sensitivity can decrease. Now it can sensitivity also varies with the type of ELISA that you are using, whether it is competitive or indirect or sandwich. All these things also sensitivity will depend. Then what type of antibodies you are using, and the specific antigen that is being targeted by the antibody present in the sample can also influence sensitivity. Now what is specificity? Specificity means they will identify the true negative. Yes, you don't have the disease, I will identify you. That is specificity. Specificity means it will identify individual who do not have the disease. Okay. So specificity means it will identify individuals who do not have the disease. So suppose you are using it for ELISA for corona whether these patients from such and such hospital have been infected with corona or not. Now those which are infected with corona, ELISA will give true result, and that is sensitivity. And ELISA has also a tendency that, not a tendency, a positive point is that it will also identify individuals who do not have the disease. So those who are not infected with the disease also can be identified by ELISA, and that is specificity. So those who do not have corona will also be shown in ELISA results. Okay, that is specificity. Now specificity again, it can be influenced by not washing. Okay, at each stage. That's why I told you, you have to remember why you are washing at each stage. If you are not blocking, if you are not washing, all these things can affect sensitivity and specificity. Now specificity means if antibodies cross react with other molecules. If you are not washing, Antibodies have a tendency to cross-react with other molecules and they will affect specificity. Okay. Now see the statements. If the plates are not blocked with BSA, I told you blocking is non-specific binding of molecules present in your sample to the free spaces in the well. Okay, to prevent that you do blocking. So if the plates are not blocked with BSA, the specificity of the assay decreases. Yes, because it will result in non-specific binding of other substances in the sample, and these can react with even your secondary antibody or even your primary antibody, and they can affect specificity. So you will not get a true negative. Sometimes a person will not be infected with corona, but since you didn't block it with BSA, there are chances that he will give a positive signal telling that he is affected with corona, but actually that is not the case. So it can affect specificity. Now if the plates are not washed between addition of serum sample and addition of anti-mouse IgG HRP, that is your secondary antibody, that is over here. If you don't wash here and you don't wash here between sample addition and addition of this secondary antibody, that is over here, if you don't wash, then sensitivity of the assay decreases. Yes, this washing is required to remove any unbound antibodies. Okay, I told you there may be certain antibodies which are freely floating in the solution that you have added, which are not bound to the antigen. So here it is unbound anti-ovalbumin IgG, and if these unbound antibodies are present, they can bind to the secondary antibody and can result in altered result. So, and it can result in reduced sensitivity also. Okay. So you may not get individuals showing true positives, that is those who are infected with corona as well as those who are not infected with corona will give the same result. That is what is meant by this sensitivity, that the sensitivity of the assay decreases. Yes, that is also correct. So A is correct as well as B is correct. So A correct, B correct, so 2 gone. Others we have to see. B, here they have not mentioned B, so that is also not correct. Okay, now see the others. So if the plates are not washed between addition of anti-mouse IgG HRP, that is over here, and the addition of H2O2 OPD, that is this washing step, the specificity of the assay decreases. Yes, that is also correct. This is because secondary antibody is designed to bind to the primary antibody, and if any unbound secondary antibody is remaining, then it will react with the substrate and it can result in false positive signal. So it will reduce the accuracy of the assay. So specificity of the assay decreases because of cross reactivity. So C is also correct. So A, B, C correct. So 1 gone. So between, okay, so 3 is the answer. Okay, now we will see the others. OPD is the substrate for the enzyme. OPD, it is actually a chromogenic substrate. It is not the exact substrate. Exact substrate is your H2O2 for HRP. Okay, the reaction with the of this chromogenic substrate and HRP, it produces a yellow-orange colored product. And a chromogenic substrate, it is a synthetic molecule, and it is designed to be in such a way that it can be acted upon by the enzyme, and it produces a visually or it produces a detectable color change. We can see with our eyes. That is what is meant by visually detectable. Okay, so that color change we can detect because of the presence of chromogenic substrate. Now to HRP you can add even another substrate, that is fluorogenic or chemiluminescent substrate instead of OPD also. You only want to detect the signal, but actually H2O2 has to be present. If H2O2 is not present, no reaction you are going to get. So OPD is the substrate for the enzyme is not correct. Then without H2SO4 no color is developed. That is wrong, because H2SO4 it is added to the enzyme reaction so that it will stop the reaction, or I can say it will stop the enzymatic reaction and thereby prevent further color development. So after 2 minutes or 1 minute, depending on the standardization that you have done for your ELISA, you can add H2SO4, and H2SO4 will change the pH of the solution. So change in pH means the enzyme activity will decrease, because the pH change can denature the HRP enzyme, and it will stop the reaction from proceeding further, and it will stabilize whatever color product has been developed. And this is required for accurate measurement. So E is also not correct. And next one. Given below is a list of microbes or their components, that is X, and specific stains Y used for their identification. Okay. Which of the following represent combination of correct matches? Now Bacillus subtilis, okay, the stain is gram stain, so A4, that makes option 3 wrong. They have given A1, so option 3 gone. Then Mycobacterium leprae, that is by Ziehl-Neelsen stains, B6, okay. So here, okay, B6, that B, option B also we have to see. Then Pseudomonas flagella is by silver stain, that is C will be 5. So that makes option 1 gone. They have mentioned C3, which is not correct. Then Volantine granules of Corynebacterium diphtheriae, it is by Albert Stein, so D1. D1, okay, there is seen in only in option 4. Okay, see the others. Cultured fungi by lactophenol cotton blue, so E5, okay, 1 is already wrong. Then Klebsiella capsule India Inc, F7, okay, F7 here they have mentioned, but that is a wrong statement. Okay, B gone. Second option also gone. They have mentioned F2. That makes option 4 as your right answer. Then Clostridium endospore, it is malachite green. So G2. So E3, yes, C5 and D1. So that makes option 4 as your answer. And these particular bacteria and or their components and what stain is used to identify them, you have to learn it by heart. Okay. Now coming to the next one. Given below are leaf length in centimeters measured from a sample of 15 dipterocarp trees. What are their mean, median, mode? Okay. Now mean is sum of the leaf length by number of values. So add up all these values. How many are there? 15. And you get 8.93. So that makes option 1 gone and option 4 also gone. Oh, sorry, not 4, option 3 gone. Okay. Now median, it is the middle value of the data when you arrange it in either ascending or the descending order. Usually it is done for ascending order because that is what is more convenient. And the middle value is taken as the median value. Now here, since it is odd numbers, you take the middle value. Okay. Now since there are 15 of them, 8th value will be the median. If it was even numbers, instead of 15 it was 16, then you take the median by adding the central 2 values and divide by 2. But here it is, luckily it is odd numbers, so the median value will be the 8th value for 15 numbers. So 9 is the answer. Okay, median 9. Okay, 2 also 9, 4 also 9. Then mode, it is the value that appears most frequently. Again you arrange them in ascending order and you find that the value which is more frequent is 8. The 7 and 9, 10, 11, all these are coming only twice. 5, 6, 12, 13 only once. So which is appearing more number of times? 8. So mode is 8, that makes 4 gone. For mode 9 they have mentioned. So answer is option 2. 8.93, 9 and 8. Then this is the flow cytometry data, and I have explained that in the morning shift also. So when I was telling on flow cytometry data analysis, I told you that each quadrant it will have a negative or a positive value, or both positive and both a negative value for the x and y axis. Okay, I will tell you that when I am discussing this question. So breast cancer cells may be identified by analyzing the CD24-44 phenotype of a breast tumor. CD24-44 stained cells were analyzed by flow cytometry, and the analysis is shown below. So this is what they got. Then they further analyzed some cells from different quadrants, that is Q1, Q2, Q3, Q4, and they immunoblotted, and this is the result what they got. So the dotted lines represent lanes with negligible signal, that is these are the dotted lines. So that means the signal intensity is very less for these particular markers. So based on the above result, which phenotype is associated with the maximum number of breast cancer stem cells? So which one is associated, you have to find. So if you see the quadrant, you can see the quadrant 1. It shows events which are positive for y-axis, that is this is your y-axis, that is CD44 plus, and x-axis is CD24 minus, so x is negative. Whereas quadrant 2 it has both x and y-axis positive, and quadrant 3 it has x and y with the negative values, and quadrant 4 it has x which is positive and y which is negative value. Okay. So I have written it over here. Here I told you x is negative, y is positive. So x value is CD24. So here CD24 will be negative, and y will be CD44. So it will be positive. Here both are positive, here both are negative, and here CD24 is positive and CD44 is negative. Now you have to see the immunoblotting results. Now before coming to that, the SOX2, OCT4, NANOG, all these are transcription factors, and in embryonic cells they are essential for maintaining pluripotency. But these cancer transcription factors, they are also found in the cancer cells, and they promote the they promote stem cells properties in the cancer cell, and it is responsible for tumor aggressiveness. So these are present at higher levels. This SOX, OCT, NA, NOG transcription factors, they are present at higher levels in the breast cancer tissues. Okay. Now what is HTERT? So HTERT is human telomerase reverse transcriptase, and it is the catalytic subunit of a telomerase. Telomerase means it will prevent shortening the ends of the chromosomes. So in cancer cells, this HTERT, it will be upregulated. It will be more, it will be expressed more, so that the cells can maintain their telomeric ends, and it will be necessary for self-renewal of these cancer cells and all that properties of the cancer cells. And beta tubulin, it is just a control. Okay, so SOX, OCT, NaNOG must be in high level in the cancer cells. Similarly, HTERT should also be expressed at high levels in the cancer cell. So when you analyze this immunoblot, you will find that Q3, Q4 it is less signal intensity. And if you see the HTERT, you can see that it is also less band intensity than that in Q1. But if you see Q2, again these transcription factor signal intensity is more in Q1 than in Q2. So that makes that phenotype associated with the maximum number of breast cancer cell is seen in the quadrant 1. And quadrant 1, you know, it is CD24 negative and CD44 positive. So that is the phenotype of the cancer cells that are associated with quadrant 1 and which are maximally expressed in your breast cancer stem cells. So that makes option 3, I suppose. Yes, CD24 minus CD44 plus as your answer. Then next one. The absorbance value of dye measured at 600 nanometers were plotted against its corresponding concentration as given below. So this is this absorbance value against concentration. Which of the following will be best estimate of the extinction coefficient of the compound? So you have to find out the value of the extinction coefficient in moles in per mole per centimeter units. And the path length of the cuvette, it is measured as 1 centimeter. So here if you see, this is the graph they have given, and how you will find from this graph the extinction coefficient. So absorbance, you know, it is epsilon CL, where and we have to find the value of the extinction coefficient, that is epsilon. And epsilon is equal to absorbance by CL. Now they have given the value of L as 1 centimeter. Concentration, you don't know. Okay. And absorbance, you don't know. So if you see the slope of this graph, if you take the slope of this graph, you will get absorbance by concentration value. Okay. So slope of this graph is delta y by delta x. And what is delta y? It is the difference in 2 points on the y axis. So y2 minus y1, and delta x is difference between 2 points on the x axis, that is x2 minus x1. So how will you find this y2 and y1 and x2 and x1? So simply just like that, take 2 points on the x axis. Here I have taken 6 and 12. You can even take 12 and 18 or 15 and 21, any 2 points. If you want, you can do it and see on your own. Okay, now I have taken 2 points. One is 6 and the other is 12. So x value will be, x1 will be 6, x2 will be 12. Now what is the corresponding y value? It is 0.2 for y1, and this is the corresponding y2 value, that is 0.4. That is what I have drawn here. 6 and 0.2 and 12 and 0.4. So here 6 is x1, y1 is 0.2, x2 and y2. So these are the values. Okay. Now you substitute it. So y2 minus y1, so 0.4 minus 0.2 and 12 minus 6. Those you are substituting here, and L, you know, it is 1 centimeter. But don't forget the unit of centimeter. It has to be there. Okay. It doesn't cancel out. So here you will get 0.1 by 3 into millimoles. Here concentration is in millimoles. So millimole per centimeter. Absorbance doesn't have any value. So 0.1 by 3 you can even write it as 10 raised to minus 1 by 3, and this you can write it as 1 by millimole per centimeter. Okay, now convert millimole to molar. So divide by 1000. So it will be written as 10 raised to minus 3. Now you take this 10 raised to minus 3 on top. So it will be 10 raised to minus 1 by 3 into 10 raised to 3. This will become mole raised to minus 1, centimeter raised to minus 1. That is what the unit for epsilon. So now you add the powers. You will get 10 raised to 2 by 3 or 100 by 3. That is 33.3 per mole per centimeter. So that is your answer. Okay, 33.3. Now next one. A molecule absorbs light at x nanometer wavelength and emits light as fluorescence at y. Okay, so one is excitation, that is x, and the other is emission, that is y nanometers. Typically there is a shift in the wavelength where y is greater than x. Okay. E is the energy transferred to the solvent during reorganization of the excited state. And H is the Planck's constant and C is the speed of light. Now E is equal to, now if you know the formula, you are studying it from your high school, E is equal to H mu, where H is the Planck constant and mu is the frequency. And frequency can also be written by the formula C by lambda, where C is the speed of light and lambda is the wavelength. So E is equal to hc by lambda. So they have asked for E is equal to, so or the change in energy, so that is delta E, that will be hc by lambda excitation minus hc by lambda emission. Hc is a constant. Okay, so substitute, excitation is x and emission is y. Hc we take out, it is a constant, so 1 by x minus 1 by y, and when you take the LCM you will get it as y minus x by xy. So answer is option 2. Okay. Then next question. A purified 150 kDa species obtained from gel filtration column. That means the protein has been obtained in a native form. Gel filtration column will give you protein in the native form. Not, what is that? Broken down into subunit form. All that you will not get from a gel filtration column. You will get it in the native conformation. Then this was run on a two-dimensional SDS page. So a similar two-dimensional SDS page has been asked in your morning shift also. I have discussed it there. That was a different question. The band position was at different places in the morning shift. You can go to my channel and see the morning session shift and you will find it there. I have explained it there. So here this is the first dimension, that is SDS page, and this is the second dimension, SDS page plus DTT. So what you have learned is first dimension isoelectric focusing, second dimension SDS page. Okay, now keep that information to one side and see what they have asked. So first dimension here is SDS page, second dimension is SDS page plus DTT. Now come to the explanation. Okay. So here before explaining this, I will tell you that first dimension, the SDS page, it will break down the weak interaction between the protein, not covalent interactions. Only weak non-covalent interactions will be broken down. Whereas when you do SDS plus DTT, DTT is the one which will break the disulfide bond between the protein. And SDS, what it will do, it will just unfold the protein by breaking this weak interaction, and it will give an overall negative charge. And the proteins, they will move on the SDS page and separate based on their molecular weight. So in this figure, when you do the first dimension, that is in this direction, you apply the sample over here. This is the wells, and you get two bands, one band over here and one band over here. Then you rotate this, then you rotate the gel, and you do the SDS DDT run. So this is the SDS page run. See here 1. So I have marked it 1, 2, 3, 4. So here when you rotate it, you get the 1 over here. 2 will be here. So these are the two bands that you got by SDS page. So this second band is what I have shaded here. Okay, here will be your sample application point. So this is the second band, and when you do the SDS DDT run, you will see that this band has moved from this position to this position. That's all. Okay. And this is the gel that they have shown in the question. So when you again rotate this one 2 times, you will get this gel. Okay. So 1, 2, 3, 4. You get 2 bands. Now did DDT act on this? No. The only thing was the second band, it got shifted from this position to this position. That's all. So what does this mean? This means that the subunits of the protein have weak interaction among themselves, or they are non-covalent interaction. That is why Upon treatment with SDS was sufficient enough to break this weak interaction. But upon treatment with SDS and DDT, we didn't get a new band. Only thing was this band got shifted from here to here. Okay. So that's it with the explanation of the gel. So SDS was more than sufficient to break the weak interaction or non-covalent interaction between the protein subunits. Since there are two bands obtained, it shows that the protein is having two subunits, or even it can be more than two also. If it is homodimer, homotetramer like that, then it can be two or more than two subunits. Okay, now whatever, they have not asked all that here, so I am not going into it. It shows that protein is having two subunits. Okay, now come to the question. So what have they asked? What is the likely form of the 1 KDA species from this observation? There are at least two proteins that are linked through non-covalent interaction. Yes, that is the answer. I already told that. It is because just by application of SDS page you got two bands. Okay, and SDS page, it breaks down the non-covalent interaction, not covalent bonds. So that makes option 2 wrong. Two proteins in the complex that are linked through covalent bond is not correct. Now, if you see the third statement, there are two proteins in the mixture without forming a complex. If that was the case, you will not get 150 KDA species upon gel filtration column. So, if there were two different proteins, then you will get two different proteins of two different molecular weight. That is what is not being shown here. Okay, you got only two bands, not two different proteins. So that means 3 is wrong. There is only one protein and it has a disulfide bond. No. DTT didn't break any disulfide bonds here. So it doesn't have any disulfide bond. Then next one. Given below are recognition sites of some restriction enzyme with the sites of restriction marked with this symbol. So EcoR5, Hintris, MA1, AVA1, SAL1, XBA1. So which of the following option represent all enzyme treated vector insert fragment combination that would generate compatible ends for ligation without any other intermediate enzymatic treatment? Now these, okay, now before coming to that, so see here, now restriction enzyme, they can create either blunt ends or sticky ends after digestion. Sticky ends means they will have a short, see here, this will create this cutting, that is so EcoR1 cutting, one cut over here and the other cut on the second strand, it will create sticky ends like this, that is they will have a small overhang which is single stranded. Okay, here also single stranded. Now when you cut the insert DNA also with EcoR1, you will get small single stranded overhangs, and when you ligate them by adding DNA ligase, because of the complementary base pairing, they will tend to ligate together. That is, this vector, DNA and the insert DNA, they have complementary base pairing in this single stranded overhang region, and they will base pair. So when you add your vector and your insert DNA, they will ligate together and you will get your insert inserted into the vector backbone. And with after you get the inserted into the vector backbone, you can go for your cloning procedures. For that only you use this restriction digestion technique, applying the complementary base pairing analysis, complementary base pairing of the DNA sequences. Okay, now this what is that? Vector and insert can be digested with those restriction enzymes which will create blunt ends. That is, they will not have this single stranded overhangs. Okay, I will show you the blunt end cutting. Now blunt end cut vector and insert DNA, they will ligate only if both are cut with the blunt end creators. That is blunt end restriction enzymes. Vector has to be cut with blunt end. Insert also has to be cut with blunt end. Only then they both can be joined, or they both can be ligated. If you cut the vector with blunt end and you cut the insert with the enzyme that will create sticky ends, then they both cannot be ligated. Okay, this is what you should know. Now usually for easy cloning, to avoid too much complications, what you do is you digest the vector and the insert with the same restriction enzyme. So this tendency or this principle of complementary base pairing is 100%. You are digesting with the same restriction enzyme. Okay, this will create a complementary end over here. It can join here and this end can join here. That is for sure. You are using the same enzyme for cutting. So you will get a ligated product. But in some studies, you may require that the vector be cut with one enzyme and an insert be cut with an other enzyme. So different enzymes, but they should be cut in, the choice of the enzyme should be such that when you cut the vector with the one enzyme, it should have a overhang which is complementary to the overhang created by the different restriction enzyme used to cut the insert. Okay, now when explaining the question, you will understand what I meant. Now see here, any complementary sticky ends can ligate. So here the vector is cut with CLA1. So it will cut over here, it will cut over here. This is the overhang that you get. Now insert is cut with BST-B1. It is a different enzyme. It will also cut here, but you see this is creating a single stranded end which is complementary to this one. So they both will ligate. So AT-TAGC can form complementary base pairing with this end CG. So AATT. Now suppose, okay, I am not going into detail why you do this cut. Okay, so for other type of studies, you do this type of cut where you want only complementary ends being bound together, whether you cut the vector with one enzyme or you cut with the insert with an other enzyme. So this is mainly based on the principle of complementary base pairing between the DNA strands. Now in the question, you have to see which all combination of these enzymes will give you the correct complementary base pairing. Okay, now see hint 3 and sol 1. So this is hint 3. They have given the sequence there in the question. So hint 3, sol 1. Okay, it will cut here, here. Hint 3 will cut here. This is the sequence that you are getting. And this is the hint 3 sol 1 sequence that you are getting. If you see here, there is no complementarity between these two sequences. AT, TCGA. It is TCGA here. How it will form complementary base pairing with this sequence? No, that is not possible. Similarly here it is AGCT. It is AGCT here. How it will form? No chance at all. So they are not compatible. That makes one gone. Okay. Now SMA1 and XBA1. So this is SMA1 and this is XBA1. So this is getting a blunt end. See here, CCC, GGG. So it is cutting over in the center here itself. You will get CCC, GGG. You won't get any overhangs. But this will create, it is cutting at the ends here. That is next to T. And here it will be the one with the next to T here. So it will create overhangs, or it is doing the staggered cut. That is what you call the cut by restriction enzyme, which is creating these cohesive ends, or which is creating the overhang ends. You call it as the staggered cut. So both are different cuts. They are not going to ligate in any way. So that makes option 2 also wrong. So 1 gone, 2 gone. Now 3, if you see hint 3 and sol 1, again the same problem. As in first one, I already explained that Hint 3 solve 1, so that makes 3 also wrong. That so your answer will be 4. Now how 4? Okay, see here, this is eco R5. Both are blunt ends. So here and small one, both are blunt ends. So CCC, GGG, GAT, AGC. So eco R1, small one. Both blunt ends, they will be compatible and they will ligate. So that is the case with this one, eco R1 and small one. Now what about AVA 1 and SAL 1? So AVA 1, they have mark given Y and R. Y means it can be any purine, and R means it can be any pyrimidine, and R means it can be any purine base. Now C, I have taken pyrimidine as thymine. Okay, C, G, and purine I am taking it as A and G, and we are cutting it. You get 2 fragments like this. And here it will generate 2 fragments with single stranded overhangs. So this will ligate with this. So CGAGCT. So AGCT means it will form complementary base pairing with TCGA. So they will form complementary base pairing and you will get a ligated end product. So that makes option 4 as your right answer. Okay, so that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CSIR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, select CSIR net exams and there you will find. If you have any doubt regarding any questions over here, you can ask it in the comment section. Thank you.